everyone, this is Dr. Sonal Jain from Department of Anthropology, University of Delhi. This is in the series of forensic science lecture. We are again discussing about various tools and techniques used in forensic sciences. There are various divisions of forensic sciences where we analyze various evidences that are categorized based on the biological evidences or the physical evidences. So whenever at any crime scene, any evidence we collect, it depends which kind of evidence we have collected, in what position or in what the type of evidence it is. It is a pattern evidence, it is a physical evidence, or it is a biological evidence. Depending upon the type of evidence, it goes to the specific division and it undergoes various specific tests depending on the nature of evidence. The schematic analysis of chemical, biological and physical samples, basically whenever we have any forensic evidence with us, firstly and foremost we have to go for the preliminary test. These preliminary tests will let us to know whether the substance which we are thinking, for example, we have collected any red substance from the crime scene. So the forensic examination might think this is blood, but the preliminary tests are required to know whether the substance which is there or the evidence which is being collected from the crime scene, it is blood or not. Then after confirming that the substance is blood, we have to go for the confirmatory test. The confirmatory test could be qualitative test or the quantitative test. Depending upon the techniques we are using, the qualitative test will tell us about which all substances are present in that. For example, there is a mixture or there is any unknown drug or any unknown substance which is being collected from the crime scene. So you have to know what is the composition of this particular substance. Let's suppose we have a white powder with us from the crime scene and we have identified using the preliminary test or using the color test that this is some drug. But what is the quality? I mean, what is the composition of that drug? So we can run various tests, for example IR, to know the composition of that drug. After knowing the composition of that drug, we have to know in how much quantity those substances or different chemical substances are present in that powder. So we have to go for various quantitative tests that can be done using mass spectrophotometry or other chromatographic techniques, can be used for both qualitative as well as quantitative tests. The first and foremost thing, whenever at a crime scene there is an evidence, evidence is being transferred from the suspect or the criminal to the victim or at the crime scene on the basis of Locard's principle. Locard's principle is the principle of exchange. So when a person comes in contact with an object or any other object comes in contact with another object or another person, there is a cross transfer of evidence which occurs. So this is the basis of forensics, that the Locard's principle, there is an exchange which occurs. For example, if we say the suspect is there or the criminal is there, his fingerprints on various places whenever he enters the crime scene, at the point of entry, or if he touches anything at the crime scene, or his fingerprints on the weapon he is carrying, all these things we have to look for that where the cross transfer might have happened. There could be the fabric, his fabric, whatever he is wearing, the threads from the shirt of the criminal or the suspect, or his footprints, his shoe prints, or anything which he is carrying, the car he is driving or any other vehicle he is driving, the tire marks of those vehicles. These all are the evidences which are being left by the suspect or the criminal at the crime scene, and these are on the basis of the Locard's principle of exchange, that they come in contact with any surface and thus the evidence was left there. Now second thing is what kind of evidence is being left, or what kind of evidence is present at the crime scene, which is being collected by the investigating officers during the crime. It depends upon the intensity, duration and nature of materials in contact. For example, if the suspect or criminal is wearing the gloves, then there are no chances of transfer of his fingerprints on any other substance or any other material or anything he touches. But his shoe prints and footprints can still be transferred to the place from where he has entered or from where he has made an exit. Similarly, the extent of transfer of footprints also depends on one thing, that whether the footprints are there in the soft mud or on the hard surface. If the footprints are on a soft mud, like any wet area or wet surface, then we can have a good 3D print of footprints which can clearly tell about the class characteristics or the individual characteristics of that particular footprint and can help in the analysis or the identification of that particular person. But if the footprint is on any hard surface and there is no mud present, then we can have a footprint which is of very low quality, and then it might be difficult for us in the lab to analyze that evidence, or for the investigating officers which are present at the crime scene to collect that evidence properly. So all the trace evidences also, if the criminal or suspect has sipped anything at the crime scene, then his or her lips marks maybe can be found at the crime scene, or supposedly there is a case of hit and run. So in such cases, the intensity of the hit to the car, from one car to the another car, the duration and the part where the both vehicles have come in contact, it all depends on the extent of the transfer, that how much the with intensity the hit and run case might have happened, and the evidence which has been left from one vehicle to the other. So all the evidences which we found at the crime scene depends on various factors: the intensity, duration, nature of materials and the extent of transfer. No crime scene is similar. Not even a single crime scene is similar to another crime scene. Every crime scene is different. It is the presence of mind of the investigating officer, that where does he look for the evidences, at all which places. So the evidences which are found at the crime scene, they could be either direct evidences or they could be indirect evidences, or we can call them as circumstantial evidences. So the direct evidences are the first hand observations. For example, the whatever information we can get from the eyewitnesses or from the video cameras, or if possible in any case if we have a confession from any suspect or any criminal, then those are the direct evidences which can be directly submitted to the court of law. But then there are indirect evidences, or we can also call them as circumstantial evidences. They usually used to imply or to prove a fact. For example, they may link a suspect and a crime scene. We know that the suspect, this is the particular person who is, this gun belongs to this suspect, and this suspect's gun was found at the crime scene. Then we can look for the indirect evidences and we can analyze that evidence in the lab. The indirect evidences composed can be categorized into two parts: physical evidences and the biological evidences. Based on the properties, we have categorized that those evidences which can be analyzed in the physics division or the ballistics division, they are the physical evidences, and the biological evidence are those evidences which are originated from the person. So otherwise the evidences which are present at the crime scene and which can be easily visible to the naked eye or can be visible using any aid like UV light, etc., all those evidences in common are known as physical evidences. But on the properties and based on where they will be analyzed, in which division they will be analyzed, they can be further categorized as physical or biological evidences. The physical evidences could be any impressions like fingerprints, footprints, the fibers from the clothes, the weapons at the crime scene, the bullets, the shell casings which are left after any round of fire has been fired by the criminal or the suspect. And the biological evidences, they make group of suspects very small or even one individual, because the biological evidence can help in direct identification of the person, but the physical evidence will lead you to the person. For example, at the crime scene we have found a bullet or a shell casing which is showing that the murder or the crime has involved a weapon using a 9mm pistol. So the 9mm pistol, if we have 10 suspects and we know that 5 suspects have 9mm pistol, so we can shortlist the list of suspects using this physical evidence that 9mm pistol was found at the crime scene and 5 suspects are having 9mm pistol, so they are under the suspect list. But another, if we have any biological evidence, if we found any biological evidence like the blood on the crime scene, then we can directly, after extracting DNA, directly relate the person with the evidence. That means the individual identification is based on the biological evidence or the fingerprints, and the physical evidence can help us in narrowing down the number of suspects of the group. So the physical evidence, we look for the chemical property for their analysis. For example, we have color, density, solubility. In the cases of if there is any burglary incident and the glass breaking is there, then we can analyze for the refractive index, things that can help in identifying something without changing the object. Those are the physical properties, but the chemical properties, for example any color change, pH change, etc., we can note that. There are certain evidences which are transient in nature and which can go away with time. For example, if there is any evidences which leave some smell or some odor, for example the perfume of a person, or in the cases of fire we have gasoline which is evaporated in nature, or any substance which can dry up, for example urine, or any burning substance, or the smoke of cigarette or cigar smoke, etc. These are the odor, or these are the substances which are transient in nature and which will go away with time. So the investigating officer should look for which evidences has to be collected first, which evidences has to be collected properly in which type of container, so that the evidence should not get lost on the way or before examination. They should also take care of those substances which are temperature dependent and which can lose their property or lose their, for example the surroundings of temperature can make any evidence bad. If any evidence which is lying outside, it's a crime scene which has happened outdoor, so any evidence, any biological evidence, for example blood, semen, it's lying outside and the temperature is very high, then there are chances that the DNA will get degraded in those such evidences. So the investigating officer first look for that which evidences has to be collected first, which evidences has to be collected in what type of container, and how the various imprints and indentations should be collected so that they do not get lost with time. The pattern evidences which are produced by the direct contact, or the evidences which are formed, for example the imprints, indentations, striation, marking, fractures, etc. The blood spatter analysis which has to be done at the crime scene to know from where the which angle the bullet was being fired to the victim and what blood spatter was being formed, to know the various angles. The glass fracture pattern, using the refractive index we can analyze the glass at the crime scene. The fire burn pattern will give us where was the starting of the fire, and which type of material was used to ignite the fire. Various furniture position can also help us in reconstructing the case, from where the victim was standing, where the suspect was present. The projectile trajectory will give us where the suspect was standing and where the victim was present, and in which direction the weapon was used, and what was the projectile trajectory from where the suspect has fired a round on the victim. Tire marks and skid marks are usually present in the cases if the suspect was traveling in a vehicle, or tire marks may be found or the skid marks may be found in the cases of hit and run accident cases. Various GSR can be found on the hands and the clothes of both suspect as well as victim. The body position is also very important to maintain, and the sketch should contain the proper body position to know at which angle the victim was being killed. Mostly this is also helpful in the cases of suicide, whether to know that the case was of suicide or the case was of murder. Various tool marks to they are usually found in the cases of burglary, to enter into the house or to open any safe, etc. And we have to look for the indentations, and they can be compared under comparison microscope. Apart from that, the evidences are categorized in the form like biological or physiological evidences which are produced from our body. They are blood, semen, saliva and other bodily fluids. The chemical substances which goes to the forensic chemistry or toxicology division are the narcotics, prescription drugs, powders associated with firearms. They also go in the ballistics division. The physical evidences could be the fingerprints, footprints, impressions, tool marks, tire marks and other firearms evidences. And the miscellaneous evidences could be the photography, dye marks and the voice analysis. So, whenever any evidence is being found, firstly we have to look for the various class and individual characteristics of these evidences. In the class evidences, a group of objects or persons with characteristic physical evidence common to it. For example, it includes the soil. So, if there is any soil marks or any soil evidences on the shoes of a person or in the clothes of the person, then we can easily say that this soil belongs to this particular area. Categorizing or explaining that this person has travelled using his vehicle or himself through this area. Individual evidences or the individual characteristics are those which can implicitly says that this person he was present at the crime scene using his fingerprints or his DNA, because that helps us in the individualization of a person. It helps us in establishing the individuality. For example, the fingerprints and DNA. So, a person can be identified or individualized using the individual characteristics, or evidence can be individualized on the basis of individual characteristics. Individual characteristics that can be associated to a common source with an extremely high degree of probability is said to possess individual characteristics, but a class characteristics evidence associated only with one group is said to have class characteristics. The class evidence is said to possess class characteristics when it can be associated only with a group and never with a single source. For example, a footprint is found at the crime scene and the footprint is of size 10. Then the suspects we are looking for, then we can classify the suspects on this, that a person who is wearing a size 10 is a suspect among the number of various suspects. That is a class evidence where the persons are being shortlisted, but only a group of people can be identified who wears a size 10 of a particular shoe. However, the high diversity of class evidence often makes it very significant. If we say that the person is wearing a size 10, of this particular brand, then that can be help us in individualizing a different person. In individual evidence, the matching ridge characteristics of two fingerprints can help us in the individualization of a person. For the individualization, we have to look for at least 9 to 11 characteristics to match a fingerprint. The comparison of striation markings on bullets or tool marks help us in individualization of an evidence. The comparison of irregular and random wear patterns in tire or footwear impressions can also lead to a person that to whom this particular car with this tire marks or this footwear impression belongs to. The comparison of handwriting characteristics, fitting together of a jigsaw puzzle, matching sequentially made plastic bags by striation marks running across the bags, all these can be helped to analyze the evidence. So firstly, the preliminary methods, they include the color test, smell test, flame test, solubility and the precipitation test. And the confirmatory methods include the microcrystal test, microscopic test, thin layer chromatography and various instrumentation like HPLC, GLC, spectroscopy, mass spectroscopy, etc. For the physical examination of blood and blood stains, we have to first go for the color test. We have to go for the physical examination. In natural light examination of exhibits for brown, reddish brown stains or the crystal of reddish brown color, these areas should be demarcated. In case of absence of clear and visible stains, wash stains should be examined under the wavelength of 230 to 269 nanometer frequency of UV light. Various presumptive tests. These suspected blood stain contaminated materials should be tested for positive for blood. So we have one tetramethyl benzidine test where we can take or swab the stain on a paper for a filter or spot test, and we can put 3% H2O2 hydrogen peroxide. If we observe a blue green color which is positive for peroxidase activity, which indicates that the hemoglobin is present in this, but this is again not a confirmatory test for blood. It could be a false positive test. So we have to go for the phenolphthalein test. The phenolphthalein test, on the swab or the cutting, we can put 2 to 3 drops of ethanol and then phenolphthalein solution is added on that particular swab. If no color is developed, then we add 3% H2O2. A intensive pink color will indicate the peroxidase activity, which again confirm the hemoglobin is present in this particular substance. But for the confirmatory test, we have to go for the crystal test. The crystal test, we have Takayama test and the Techman test. In the Takayama test, the material which has to be tested, we will take a slide and cover it with a cover slip, putting a drop of material which is to be tested. A Takayama reagent, drop of that is allowed to flow under the cover slip. For the test to occur, we have to slightly warm the slide at 65 degrees Celsius for 10 to 20 seconds, and then we can observe under the magnification of 100x in the microscope, in the normal compound microscope, and the presence of pink needle shaped crystals which are formed because of the pyridine ferroprotoporphyrin gives us a positive reaction for heme, confirming that this is a confirmatory test for blood and the blood is the material which we are looking for. Similarly, if we examine various semen and seminal stains, the physical examination will show that this is a thick yellowish white liquid and it has an odor. The smell will tell us that it might contain semen. Then we can look for the texture of that. Texture can be, the seminal stains are starchy, so we can on touching can tell us that this is a seminal stain. The appearance, looking for the seminal stain or various garments or various clothes, we can use a UV light to look for the various stains. And in natural light, sometimes the stains appear reddish colored, otherwise they are usually yellow or faint grey in color and can easily be visualized using a UV light. They can also be mixed with the stains of vaginal discharge or urine or blood, and they can be identified using the fluorescence of the seminal stain, which gives a bluish white color, and then those stains could be examined from the clothing, and this should be sent for the further examination. We have acid phosphatase test for the semen stains. A positive reaction with sodium alpha naphthyl phosphate will give us a purple color. The confirmatory test will include the gram positive protein will stain violet, gram negative protein will stain red, and the appearance of spermatozoa will confirm that it's semen. So, the forensic techniques contribute to crime prevention and the development of investigative strategy by analyzing patterns in crime scenes behavior or modus operandi. The forensic experts can help law enforcement agencies. They can identify potential serial offenders, predict future criminal activities, and establish proactive measures to prevent such crimes. Overall, a good method or a good way for the forensic techniques are vital in gathering, analyzing, and interpreting evidence in a systematic and scientific method. This will provide critical information that helps in solving crimes, supporting legal proceedings, and that will also ensure that justice is well served. Thank you.